Hi, welcome to this video where I'm going to talk about some things that might possibly come up on the AQA A-level biology exam, and that's in 2024. So the first thing I want to say is you really shouldn't actually rely on this video too much. Hopefully you are feeling pretty confident overall with your revision, and as I'm putting this out just like about a week before your exam, this might just give you the final few things to really make sure you know before the exams. So first of all, a little bit about the process. So what I've done is I've gone through the past papers for the last 3 years, and I've cross-referenced them mainly with the textbook pages of a textbook that I use, the AQA textbook, and I've also looked at the required practicals. And I find this a little bit more useful than going through the spec, because the spec, especially for AQA, each individual spec point is not very precise. It's quite broad, and the textbook actually is a little bit more detailed. So let's get into it. So first of all, this is the year 12 content from the textbook. And this is all the year 12 content, so that's modules 1, 2, 3, 4, and also the first 6 required practicals that I put in down here. So that's enzymes, mitosis, calculating mitotic index, the osmosis practical looking at like pieces of potato or plant tissue and whether it gains or loses mass. Membranes, that's the beetroot practical with betalain leaking out of the vacuole inside the beetroot cells. And then also heart dissection, RP5, and microorganisms or antibiotic resistance in microorganisms, that's RP6. So let's look at what's come up over the last couple of years. So first of all, in 2023, I've highlighted everything that's come up here in yellow. So already we can see that in each year, they can't test you on everything, so they've got to kind of choose things on a bit of a cycle. If we look at the year 13 content, there's the year 13 content. You can pause here and have a look if you want. So this stuff is the least likely to come up again. Now if we go into the 2022 paper, so this is the year 12 content. So this might be worth pausing and having a look at. If it came up in the last 2 years, it's pretty unlikely to come up again, but there are some gaps here that are starting to become more apparent. So pause if you want, and now let's look at the year 13 content. So this is the year 13 content for the last 2 years, what's come up and where the gaps which haven't been assessed are. Now we can look at the 2021 paper, so that's the last 3 years, and this now begins to get quite interesting because there do seem to be some gaps that haven't come up. For example, up here we've got introduction to biological molecules. Carbohydrates haven't come up that much in terms of monosaccharides and disaccharides. Polysaccharides have, but monosaccharides and disaccharides not. Water hasn't come up for a while. What else? Haven't had so much on active transport or co-transport actually. Within immune system, perhaps B lymphocytes and humoral immunity, and maybe how antigen presentation kind of links to that. Section 3, there's some big gaps here. Look at this. This hasn't come up for so long. The human gas exchange system. So I think you really need to make sure that you know your lungs and things to do with like FEV1. That's a way of kind of diagnosing respiratory problems, so being able to analyze those graphs, FEV1 graphs. Also maybe just describing inhalation and exhalation. Digestion, absorption hasn't come up that much. Within this, it's come up quite a lot, but cardiac cycle actually hasn't come up. So perhaps something might come up this year, which is one of those kind of classic analyzing the heart pressure graph type questions, where you can say which valves are opening and closing at what points and why they're opening and closing. Protein synthesis, transcription and splicing hasn't come up for a while. Translation came up in 2021, but transcription and splicing might be something that comes up. And then maybe measuring biodiversity or in that one. Let's look at the year 13 content. Here we go. So again, gaps, oxidative phosphorylation, perhaps. This, I think, is a big area. So artificial fertilizers and everything surrounding them. So how do they work? Environmental issues. Really maybe thinking that eutrophication could come up. Because actually mycorrhiza have come up. That's the kind of fungal filaments that associate with plant roots and help absorb minerals. But eutrophication is a prediction of mine, a big prediction. Reflex arc, perhaps, possibly. And then actual kind of straightforward action potential questions haven't come up for a little while. We've had synapse, but we haven't had action potential. And actually, we have had structure of skeletal muscle. I think that's a mistake. That has come up actually last year. Maybe general principles of homeostasis and feedback mechanisms. Maybe you could have an application question about thermoregulation might come up this year. And then looking at the kidney stuff that has come up recently, actually the glomerulus hasn't come up. So that's ultrafiltration. Within inheritance, these are things that have come up, but autosomal linkage and epistasis haven't been tested for a little while, so I think make sure you really know your autosomal linkage. Down here, populations and evolution, maybe different forms of selection on evolution, and actually I think competition and predation could come up in some way, possibly linked together. So maybe the effect of competition and the effect of predation on biodiversity part, possibly. This could be maybe a paper 3 thing. So you could link these two with maybe this, possibly. This is a possibility. Going into section 8, a lot of that came up last year, actually. Section 8 was really, really heavily assessed in the 2023, but it might come up again, potentially. And then this last one, recombinant DNA technology. Things that haven't come up, potential. Actually, it's all come up quite recently. Maybe PCR, because that came up in 2021. So PCR might come up. Who knows? In terms of the required practicals, you can see which ones haven't come up for a few years. Okay, so RP8, DC PIP, RP11, glucose testing, so that's glucose testing in urine, and RP12 type questions to do with ecology haven't come up. I think I forgot to mention ones from year 12 content. It's RP1 enzymes and RP4 membranes. Okay, so that's the kind of content that's come up. I'm just going to quickly run through, again, possibilities briefly. Now, first of all, there are some things that come up again and again for paper 1. So these are the things that you just really need to know because they basically come up every single year. So protein structure, so that's talking about primary through to quaternary structure. And then how a mutation affects the protein structure. So maybe you could like research, for example, an example of that just to kind of master that content. And it's always something you could use for essays as well. So maybe you look at cystic fibrosis or sickle cell anemia and talk about how a single mutation can affect protein structure and function. Enzyme action, you've got to know everything about that. And then all of cell structure. This is really the fundamentals of all of biology. So you need to know about your cell structure. And actually, over the last couple of years, I don't think prokaryotic cells have come up that much. Cell structure. Actually, no, that came up last year. What did I think was coming up this year? I had a feeling that mitochondria might come up because chloroplast came up, nucleus has come up, but maybe the mitochondrial structure hasn't come up. So maybe that could be something. Right, things that haven't come up in a while. Human gas exchange and ventilation. Top prediction. Got to know that absolutely like the back of your hand. And also stuff to do with what's it called? FEV1. So this kind of graph where you're looking at that, and that's like the total lung volume, and FEV1 is how much you would exhale in 1 second, and then whether like if it's kind of this sort of thing or if it's this sort of thing, like the difference between obstructive and restrictive lung diseases. Plant gas exchange and limiting water loss, so that could be xerophytes within that. Carbohydrates, monosaccharides and disaccharides. Now that could be nicely linked to food tests for monosaccharides and disaccharides, so you should know your reducing and non-reducing sugar tests. Cardiac cycle, that kind of classic graph. I'm going to very quickly sketch it, where it kind of looks a bit like this, and then you've got the aorta pressure that kind of looks like this, like that. Can you identify when things are opening and closing, basically? Membrane structure. Now this would link nicely to RP4, factors that affect membrane permeability. So what types of molecules can get through membranes through simple diffusion? What things need like protein channels or carrier proteins, and then kind of really just understanding that RP4 betalin practical that you should have done for your practical component. Transcription and splicing, RP1 enzymes. I mean, there are always enzymes questions, so just knowing this is going to be useful for any kind of enzyme practical question. And then these two things haven't come up in the last 2 years. But I have got a feeling that they might come up again. So DNA replication, maybe the measles and Stahl experiment, and then just viral replication. And this is actually T cell or cell mediated immunity. So that's cytotoxic T cells. That hasn't come up in a little while. Antibodies actually has, but cell mediated immunity hasn't come up for a little while. So when I say these are possibilities for paper 1, they are of course also possibilities for paper 3, because paper 1 and paper 3, it could come up in either one. Paper 3 is the unified any year 12 or year 13 content. Paper 2, stuff that always comes up. It seems like every single year they've got a Hardy-Weinberg question. It might just be 2 marks, maybe 3, but make sure you get those marks. So know how to do your Hardy-Weinberg, and really make sure you know the fundamental monohybrid and dihybrid crosses, genetic crosses, because that always comes up. And remember, this year specifically, I've got a feeling epistasis or autosomal linkage might be the types of crosses that you have to do. Stem cells comes up again and again. It's really common, actually, I think, in the unified paper 3, because stem cells are a really great way to kind of draw in multiple things together. You could have, like, stem cells to fix a certain disease which affects a certain organ. I don't know, maybe this year it could be something like how stem cells can be used to, I don't know, cure some sort of kidney disease that focuses on the glomerulus, because that's like several of those things I think might be coming up. Cancer and epigenetics, again, cancer is a useful type of question that the examiners like to ask, because you can bring in lots of different things with cancer. Often, cancer, gene expression, cell division, cell cycle, all that stuff links together as often a paper 3 type thing as well. Investigating populations, so that's quadrats basically. Well, that's plant populations and plant biodiversity, or capture and then recapture sampling. So you catch it, you mark it, you catch it again, and use a formula to work out the population size. Natural selection, that always comes up every year. Make sure you can kind of get your standard 4 or 5 marks for a natural selection question. So that's mutation creates a new allele. Allele affects survival, increases chances of survival. You might be able to explain why that is depending on the allele. So therefore, the allele is passed on to the next generation. You get a change in allele frequency, and you might say something like this is a directional selection that causes an increase in allele frequency. Okay, things that haven't come up in a while: oxidative phosphorylation and chemiosmosis has not come up in a while, and I've kind of got a feeling that maybe mitochondria structure might come up as well. So that would kind of link together. Use of artificial fertilizers and eutrophication. So eutrophication, when fertilizers run off into bodies of water like rivers and lakes and even the sea, and how you get this cascading sequence of changes that results in death of everything and lack of oxygen. The action potential, how it travels, potentially that could be linked to a reflex arc. Within the kidney, I've got a feeling that the glomerulus and ultrafiltration might come up this year. I haven't had many questions on that within the kidney at all. We already said epistasis and autosomal linkage could be things that come up within genetic crosses, and maybe how you determine whether something is linked or not using chi-squared. Remember, for AQA you don't actually have to calculate chi-squared, but you might need to say that you would use chi-squared to look and see if there's a statistical difference between observed values and expected values. So expected values if there's no linkage, but then the values that you actually observe are a bit different than what you expect. And then you can calculate chi-squared to see if it's above the critical value. Competition and predator-prey interactions, they could be linked together. So competition between, so interspecific competition between 2 different species. I have a weird feeling about squirrels this year. Red and grey squirrels, I'm thinking, could come up. And maybe predator-prey interactions. Actually, a colleague of mine has had a bit of a suspicion that maybe something to do with red and grey squirrels and the interaction. There's a predator called the pine marten. So just a brief bit on red and grey squirrels. Grey squirrels are introduced in the UK. They came from the United States, North America. Red squirrels are native to the UK. Grey squirrels, very strong competitors, kind of out-competing the reds. And so the reds are in danger. They've retreated to the north of Scotland and so on. But part of this is because a natural predator called the pine marten has been removed. So potentially if they, I don't know, you might have some sort of data on, reintroduction of pine martens and how this affects the grey population, because they eat the greys more than the reds, and then how that affects competition between the greys and the reds. I don't know, maybe squirrels. If it does come up, I'll be very happy. Required practicals, RP8 DC pip. So this is looking at the activity of the chloroplast. This is looking at the light-dependent reaction. And DC pip, remember, goes from blue to colorless as it's reduced by the activity of the electron transport chain in the chloroplast. RP11, testing urine for glucose. So remember I said that also I think monosaccharides and disaccharides, and that kind of test hasn't come up for a while. So this could be linked in. So how to do, a Benedict's test to test for glucose in urine kind of like brings those two things in. It brings in monosaccharides, it brings in testing for glucose, and this RP11. And then RP12, ecological sampling, could also possibly come up. Okay, so on to the essay, the last part of paper 3, the 25 marker, something that students often are afraid of. This is even harder to predict. So first of all, let's just look at everything that's come up in the past. On the right-hand side here, the yellow, this is everything that's come up since the new spec. So that's the specimen paper in 2015. Then in 2016, there was no specimen paper. Then 17 to 23. Here are all the essay titles. So looking at these titles and looking actually at some of the titles that came up in the old spec, there are a few things that maybe could come up. And actually, if you prepare for all of these, then no matter what comes up, you should have paragraphs that you can kind of shuffle around and use, but just make sure that you've got a bit of an idea for these ones. The importance of water and the regulation of water content. This is like probably my top prediction. I did think it was going to come up last year. It didn't, but that could mean it comes up this year. So thinking about how the properties of water link to its various roles, for instance, in transport systems, for instance, in the xylem, for instance, in its role in providing thermostable habitats and in thermoregulation. And then you could also talk about osmoregulation in the kidneys, etc. The importance of negative feedback. So this could be, again, temperature regulation, osmoregulation, glucose regulation, but then also in things like when a population of organisms is growing, how you get sort of negative feedback, which means that it grows more slowly and slowly and slowly and reaches its carrying capacity. The importance of receptors. So we're talking about kind of, for example, the insulin receptor, T cell receptors, B cell receptors, acetylcholine receptors, lots of different types of receptors you can talk about. And then you could talk about receptors like the Pacinian corpuscle or photoreceptors. Those are also kind of cell type receptors. 4. This is another strong prediction. The importance of nucleotides and molecules derived from nucleotides. So I feel like they haven't had much on DNA recently. In 2019, they did have this one, the importance of DNA as an information-carrying molecule and its use in gene technologies. Could come up again, I suppose, but they might just slightly spin it and have it on nucleotides. So this would still leave you able to talk about DNA, RNA, that's mRNA and tRNA. But you could also talk about things like NAD and FAD. Those are derived from nucleotides. Or cyclic AMP or NADP, are all derived from nucleotides. Okay, number 5. The importance to humans of the control of growth, reproduction, and development. That would be an interesting one. So you could talk about cell cycle. You could talk about cancer and regulating the cell cycle. You could talk about reproduction and meiosis and like sources of variation. And development. I don't know, what would you talk about in development? I suppose you talk about transcription factors. You can talk about estrogen, control of puberty, for example. So that's a possibility. And then finally 6, I was trying to keep it to 5, and then I added another one. The importance of proteins and their structure and the control of processes. This would be an easy one. Proteins are absolutely everywhere in biology. Most of the things that you've learned about, most of the molecules that you've learned about are probably proteins, but maybe have a few key proteins that you know about, like hemoglobin would be a good one. Maybe, I don't know, ATP synthase would be another good one. Lots of proteins you can talk about. Maybe actin and myosin. That would be another good one. What else? Maybe some enzymes like amylase, for example. Okay, so what to do next? Haven't got long until your exam, so focus on those predicted areas. You might want to do some full practice papers. I got a couple walkthroughs, 2018 and 2019 on the channel. And you might also want to just like fine-tune your exam technique. There's also an exam technique run through. All the best with your final revision and best of luck in your exams. Thanks.